Pollination traps or trap flowers are plant flower structures that aid the trapping of insects, mainly flies, so as to enhance their effectiveness in pollination. The structures of pollination traps can include deep tubular corollas with downward pointing hairs, slippery surfaces, adhesive liquid, attractants, often deceiving the insects by the use of sexual attractants rather than nectar reward, and therefore termed as deceptive pollination, flower closing, and other mechanisms. Arum with trap chamber at base. In many species of orchids, the flowers produce chemicals that deceive male insects by producing attractants that mimic their females. The males are then led into structures that ensure the transfer of pollen to the surfaces of the insects. Orchids in the genus Pterostylis have been found to attract male fungus gnats with chemical attractants and then trap them using a mobile petal lip. The general observation of insects being trapped and aiding pollination were made as early as 1872 by Thomas Frederick Cheeseman and did not go unnoticed by Charles Darwin, who examined the adaptations of orchids for pollination. Slipper orchids have smooth landing surfaces that allow insects to slide into a container from which a window of light leads the insect outwards through a narrow passage where the pollen transfer occurs. The structures found in large flowers such as those of Rafflesia and some Aristolochia are also evolved to attract and trap pollinators. Cirripegia rhynchantha, another trap flowers that produce deceptive sexual chemicals to attract insects, may often lack nectar rewards. Many species of arum trap pollinators and the specific mechanisms vary with the insects involved. Plants in the genus Cirripegia attract small flies in the families Miliciidae, Chloropodidae, and Ceratopagonidae, and pollinaria attach to their proboscis. An analysis of the scents emitted by Cirripegia dolichophila showed the presence of spiroacetals, which are rare in plants and common among insects. Melichid flies, which are kleptoparasites of arthropod predators, are attracted by these chemicals and become the pollinators of these plants.